Hey guys, welcome to another high-yield explainer. Today, we're zooming way down into the microscopic world of bacteria. Look, if you're an MBBS student prepping for microbiology exams, you already know bacterial morphology is an absolutely heavy-hitting topic. It's dense, but don't worry, we're going to break this all down into a super structured, highly visual map to help you secure those top marks.
I want to challenge you for a second. Ditch the heavy, dense textbooks right now. Really, how do you perfectly recall bacterial anatomy when you're sweating it out in the exam hall? Well, the secret isn't reading the exact same paragraphs over and over again. It's visual and structured learning. By the end of this explainer, you'll be able to literally map out a bacterium from the outside in completely from memory.
Here's our game plan today. We're taking a logical, outside-in journey, starting with bacterial shapes, moving into the anatomy of the cell wall, crossing the cell membrane, and finally exploring the cytoplasmic matrix. Let's dive right in.
Starting with Section 1, Bacterial Shapes. Think of bacteria as falling into two main camps based on their shape. You've got your Cocci, which are basically oval or spherical, berry-like cells, and then you have your Bacilli, which are your rod-shaped cells. And the cool thing is, they don't just float around at random. They actually arrange themselves in very specific patterns, stuff like clusters, chains, pairs, or even these wild Chinese letter shapes. These structural arrangements, especially when you combine them with whether they're gram-positive or gram-negative, form the absolute foundation of bacterial identification.
So here's where we hack the exam. You need to memorize these through association, because examiners literally love testing these exact pairings. Take a mental snapshot right now. When you read Staphylococcus, your brain must immediately shout clusters. Streptococcus, think chains. If a clinical vignette suddenly mentions lanceolate-shaped pairs, boom, you're dealing with pneumococcus. Bamboo stick appearance, that is the classic hallmark for Bacillus anthracis. And that Chinese letter or cuneiform pattern is an absolute dead giveaway for Corynebacterium diphtheria. Lock these in, guys. They are guaranteed points.
Moving one layer deeper, Section 2, Anatomy of the Cell Wall.
To really understand this cell wall, you have to understand its core building block, peptidoglycan. Think of peptidoglycan as the bricks and mortar of the entire bacterial wall. It's a mucopeptide chain made of alternating units of N-acetylmuramic acid, we're just going to call it NAM, and N-acetylglucosamine, or NAG. This exact structure is what gives the bacteria its extreme rigidity and prevents osmotic lysis. Simply put, if this wall breaks, the bacteria dies.
And this sets up the foundation for probably the most famous battle in all of microbiology, gram-positive versus gram-negative cell walls. Both sides use peptidoglycan, but how they utilize it is vastly different. This difference is exactly why the gram stain actually works, and trust me, it's a super popular topic for essay questions.
You can see the massive visual contrast between these two walls. The gram-positive wall is essentially a giant, thick fortress of peptidoglycan. We are talking 50 to 100 layers thick, spanning 16 to 80 nanometers. It also has teichoic and lipoteichoic acids woven right through it. But then look at the gram-negative wall. It's much more complex, but incredibly thin. Its peptidoglycan layer is barely 2 nanometers thick, and it's hiding underneath a lipid-rich outer membrane packed with lipopolysaccharides or endotoxins. So thick and simple versus thin and complex.
Let's zoom way in to the molecular level of that gram-positive peptidoglycan. See how the NAM and NAG alternate, but pay close attention to how they connect. Descending from the NAM molecule is a tetrapeptide side chain containing L-lysine. Crucially, the L-lysine of one chain is covalently linked to the D-alanine of an adjacent chain through a very specific bridge. That bridge is made of pentaglycine. This pentaglycine bridge is a massive, high-yield defining feature of gram-positive walls.
Now, switch gears and compare that to the gram-negative peptidoglycan. Notice what's missing? Yup, no pentaglycine bridge. It is completely absent. Instead, those tetrapeptide chains are directly linked to each other. And here's the tricky part. If you look closely at the third position of that side chain, the L-lysine has actually been replaced by mesodiaminopimelic acid. It's a really subtle biochemical swap, but I promise you, examiners will test you on it.
Okay, pause for a second. This right here is your golden ticket. If you get an essay or a short note asking you to differentiate gram-positive and gram-negative cell walls, reproducing this exact table will absolutely guarantee you maximum points. Thick versus thin peptidoglycan, L-lysine versus mesodiaminopimelic acid, the presence or absence of that pentaglycine bridge, the teichoic acid, and the outer membrane with its endotoxic LPS. Seriously, screenshot this in your mind right now.
All right, Section 3, the cell membrane.
So pushing past the rigid cell wall, we hit the bacterial cell membrane. It's tiny, just 5 to 10 nanometers thick, and follows the classic fluid mosaic model, a bilayered phospholipid with embedded integral and peripheral proteins. But here is the high-yield trick you need to know. Unlike human eukaryotic cell membranes, bacterial membranes completely lack sterols, like cholesterol. Mycoplasma is literally the only exception, and that's just because it lacks a cell wall altogether. So, what do normal bacteria use instead of cholesterol to stay stable? They use pentacyclic sterol-like molecules called hopanoids.
When an exam asks you for the functions of the cell membrane, I want you to immediately think of the acronym OTRR. O for osmotic barrier. It's selectively permeable. T for transport system, using proteins for taking in nutrients and dumping waste. R for respiration. Remember, bacteria don't have mitochondria, so the membrane handles those crucial metabolic processes. And the second R is for receptors, which act like tiny antennas helping the bacteria detect chemicals around them. OTRR. Simple, fast, and totally exam ready.
Finally, Section 4. The cytoplasmic matrix.
Now notice what's intentionally missing inside the bacteria here. Unlike our own eukaryotic cells, the bacterial cytoplasm doesn't have any of those fancy membrane-bound organelles. No rough ER, no Golgi apparatus, definitely no mitochondria. Instead, this cytoplasmic matrix, which is roughly 70% water, is basically just a bustling factory floor packed with free-floating ribosomes, storage inclusions to hold on to nutrients, and specialized membrane invaginations called mesosomes.
Let's talk about those ribosomes for a second, because they're the sites of protein synthesis. For exams, you absolutely have to know your bacterial ribosome math. Eukaryotes have 80S ribosomes, but bacteria rock the 70S ribosomes. And in the weird, nonlinear world of Svedberg units, 70S is actually made up of a 30S subunit and a 50S unit. Yep, 30 plus 50 equals 70 in bacteriology. Write it down exactly like that, and you will never confuse it with the eukaryotic ones. Next up are the mesosomes. These are basically vesicular invaginations of the plasma membrane, and you'll mostly see them in gram-positive bacteria. But what do they actually do? Just drop the RCC acronym to quickly lock in their functions. R for respiration, they hold respiratory enzymes, essentially acting as a substitute for mitochondria. C for cell wall formation, and the second C is for chromosome replication during cell division. RCC. Boom. Done. And finally, sitting right in the middle of all this cytoplasm is the genetic material. Bacteria don't bother with the true nucleus. There's no nuclear membrane, no nucleolus. Instead, their genetic material just sits completely exposed in this irregularly shaped region we call the nucleoid. It's pretty much just a single haploid chromosome made of supercoiled, circular, double-stranded DNA. And when it's time to multiply, it just divides by simple binary fission. No complex mitosis to worry about here.
So, there we have it. We've successfully built our bacterium, from its outer shape, through the tough cell wall, past the security of the membrane, and right down into the DNA. You now have a solid visual map to rely on for your exams. But that brings us to a pretty provocative question. We know exactly how they're structured, but how do these microscopic entities actually swim around their host? And when all their nutrients dry up, how do they manage to survive extreme starvation and boiling temperatures? Well, the answers to that lie in flagella and spores, but we'll save that topic for our next explainer. Keep reviewing this visual map, trust your mnemonics, and good luck crushing your exam prep.